Figure 3.1. Microorganisms vary visually in their size and shape, as can be observed microscopically. But they also vary in invisible ways, such as in their metabolic capabilities. Credit AEF, modification of work by Centers for Disease Control and Prevention. Credit B, modification of work by NIAID. Credit C, modification of work by CSIRO. Credit D, modification of work by Microscopic World, YouTube. Chapter Outline 3.1. Spontaneous Generation 3.2. Foundations of Modern Cell Theory 3.3. Unique Characteristics of Prokaryotic Cells 3.4. Unique Characteristics of Eukaryotic Cells. Life takes many forms, from giant redwood trees towering hundreds of feet in the air to the tiniest known microbes, which measure only a few billionths of a meter. Humans have long pondered life's origins and debated the defining characteristics of life, but our understanding of these concepts has changed radically since the invention of the microscope. In the 17th century, observations of microscopic life led to the development of the cell theory, the idea that the fundamental unit of life is the cell, that all organisms contain at least one cell, and that cells only come from other cells. Despite sharing certain characteristics, cells may vary significantly. The two main types of cells are prokaryotic cells, lacking a nucleus, and eukaryotic cells, containing a well-organized, membrane-bound nucleus. Each type of cell exhibits remarkable variety in structure, function, and metabolic activity. Figure 3.1. This chapter will focus on the historical discoveries that have shaped our current understanding of microbes, including their origins and their role in human disease. We will then explore the distinguishing structures found in prokaryotic and eukaryotic cells. Learning objectives. By the end of this section, you will be able to explain the theory of spontaneous generation and why people once accepted it as an explanation for the existence of certain types of organisms. Explain how certain individuals, Van Helmont, Reddy, Needham, Spallanzani, and Pasteur, tried to prove or disprove spontaneous generation. Clinical focus. Part 1. Barbara is a 19-year-old college student living in the dormitory. In January, she came down with a sore throat, headache, mild fever, chills, and a violent but unproductive, i.e., no mucus, cough. To treat these symptoms, Barbara began taking an over-the-counter cold medication, which did not seem to work. In fact, over the next few days, while some of Barbara's symptoms began to resolve, her cough and fever persisted, and she felt very tired and weak. What types of respiratory disease may be responsible? Jump to the next clinical focus box. Humans have been asking for millennia, where does new life come from? Religion, philosophy, and science have all wrestled with this question. One of the oldest explanations was the theory of spontaneous generation, which can be traced back to the ancient Greeks and was widely accepted through the Middle Ages. The theory of spontaneous generation. The Greek philosopher Aristotle, 384 to 322 BC, was one of the earliest recorded scholars to articulate the theory of spontaneous generation, the notion that life can arise from non-living matter. Aristotle proposed that life arose from non-living material if the material contained pneuma, spirit, or breath. As evidence, he noted several instances of the appearance of animals from environments previously devoid of such animals, such as the seemingly sudden appearance of fish in a new puddle of water. 1. This theory persisted into the 17th century, when scientists undertook additional experimentation to support or disprove it. By this time, the proponents of the theory cited how frogs simply seemed to appear along the muddy banks of the Nile River in Egypt during the annual flooding. Others observed that mice simply appeared among grain stored in barns with thatched roofs. When the roof leaked and the grain molded, mice appeared. Jan Baptista van Helmont, a 17th-century Flemish scientist, proposed that mice could arise from rags and wheat kernels left in an open container for 3 weeks. In reality, such habitats provided ideal food sources and shelter for mouse populations to flourish. However, one of van Helmont's contemporaries, Italian physician Francesco Redi, 1626 to 1697, performed an experiment in 1668 that was one of the first to refute the idea that maggots, the larvae of flies, spontaneously generate on meat left out in the open air. He predicted that preventing flies from having direct contact with the meat would also prevent the appearance of maggots. Reddy left meat in each of 6 containers, figure 3.2. 2 were open to the air, 2 were covered with gauze, and 2 were tightly sealed. His hypothesis was supported when maggots developed in the uncovered jars, but no maggots appeared in either the gauze-covered or the tightly sealed jars. He concluded that maggots could only form when flies were allowed to lay eggs in the meat, and that the maggots were the offspring of flies, not the product of spontaneous generation. Figure 3.2. Francesco Reddy's experimental setup consisted of an open container, a container sealed with a cork top, and a container covered in mesh that let in air but not flies. Maggots only appeared on the meat in the open container. However, maggots were also found on the gauze of the gauze-covered container. In 1745, John Needham, 1713 to 1781, published a report of his own experiments, in which he briefly boiled broth infused with plant or animal matter, hoping to kill all pre-existing microbes. 2. He then sealed the flasks. After a few days, Needham observed that the broth had become cloudy, and a single drop contained numerous microscopic creatures. He argued that the new microbes must have arisen spontaneously. In reality, however, he likely did not boil the broth enough to kill all pre-existing microbes. Lazzaro Spallanzani, 1729 to 1799, did not agree with Needham's conclusions, however, and performed hundreds of carefully executed experiments using heated broth. 3. As in Needham's experiment, broth in sealed jars and unsealed jars was infused with plant and animal matter. Spallanzani's results contradicted the findings of Needham. Heated but sealed flasks remained clear, without any signs of spontaneous growth, unless the flasks were subsequently opened to the air. This suggested that microbes were introduced into these flasks from the air. In response to Spallanzani's findings, Needham argued that life originates from a life force that was destroyed during Spallanzani's extended boiling. Any subsequent sealing of the flasks then prevented new life force from entering and causing spontaneous generation. Figure 3.3. Figure 3.3. A. Francesco Reddy, who demonstrated that maggots were the offspring of flies, not products of spontaneous generation. B. John Needham, who argued that microbes arose spontaneously in broth from a life force. C. Lazzaro Spallanzani, whose experiments with broth aimed to disprove those of Needham. Check your understanding. Describe the theory of spontaneous generation and some of the arguments used to support it. Explain how the experiments of Reddy and Spallanzani challenged the theory of spontaneous generation, disproving spontaneous generation. The debate over spontaneous generation continued well into the 19th century, with scientists serving as proponents of both sides. To settle the debate, the Paris Academy of Sciences offered a prize for resolution of the problem. Louis Pasteur, a prominent French chemist who had been studying microbial fermentation and the causes of wine spoilage, accepted the challenge. In 1858, Pasteur filtered air through a gun cotton filter and, upon microscopic examination of the cotton, found it full of microorganisms, suggesting that the exposure of a broth to air was not introducing a life force to the broth, but rather airborne microorganisms. Later, Pasteur made a series of flasks with long, twisted necks, swan neck flasks, in which he boiled broth to sterilize it. Figure 3.4. His design allowed air inside the flasks to be exchanged with air from the outside, but prevented the introduction of any airborne microorganisms, which would get caught in the twists and bends of the flasks' necks. If a life force besides the airborne microorganisms were responsible for microbial growth within the sterilized flasks, it would have access to the broth, whereas the microorganisms would not. He correctly predicted that sterilized broth in his swan neck flasks would remain sterile, as long as the swan necks remained intact. However, should the necks be broken, microorganisms would be introduced, contaminating the flasks and allowing microbial growth within the broth. Pasteur's set of experiments irrefutably disproved the theory of spontaneous generation and earned him the prestigious Alhambert Prize from the Paris Academy of Sciences in 1862. In a subsequent lecture in 1864, Pasteur articulated omne vivum ex vivo, life only comes from life. In this lecture, Pasteur recounted his famous swan neck flask experiment, stating that life is a germ, and a germ is life. Never will the doctrine of spontaneous generation recover from the mortal blow of this simple experiment. 4. To Pasteur's credit, it never has. Figure 3.4a. French scientist Louis Pasteur, who definitively refuted the long-disputed theory of spontaneous generation. B. The unique swan neck feature of the flasks used in Pasteur's experiment allowed air to enter the flask, but prevented the entry of bacterial and fungal spores. C. Pasteur's experiment consisted of 2 parts. In the first part, the broth in the flask was boiled to sterilize it. When this broth was cooled, it remained free of contamination. In the second part of the experiment, the flask was boiled, and then the neck was broken off. The broth in this flask became contaminated. Credit B. Modification of work by Wellcome Images /Wikimedia Commons. Check your understanding. How did Pasteur's experimental design allow air, but not microbes, to enter, and why was this important? What was the control group in Pasteur's experiment, and what did it show? Learning objectives. By the end of this section, you will be able to explain the key points of cell theory and the individual contributions of Hooke, Schleiden, Schwann, Remak, and Virchow. Explain the key points of endosymbiotic theory and cite the evidence that supports this concept. Explain the contributions of Semmelweis, Snow, Pasteur, Lister, and Koch to the development of germ theory. While some scientists were arguing over the theory of spontaneous generation, other scientists were making discoveries leading to a better understanding of what we now call the cell theory. Modern cell theory has 2 basic tenets. All cells only come from other cells. The principle of biogenesis. Cells are the fundamental units of organisms. Today, these tenets are fundamental to our understanding of life on Earth. However, modern cell theory grew out of the collective work of many scientists. The origins of cell theory. The English scientist Robert Hooke first used the term cells in 1665 to describe the small chambers within cork that he observed under a microscope of his own design. To Hooke, thin sections of cork resembled honeycomb, or small boxes or bladders of air. He noted that each cavern bubble or cell was distinct from the others, figure 3.5. At the time, Hooke was not aware that the cork cells were long dead and, therefore, lacked the internal structures found within living cells. Figure 3.5. Robert Hooke, 1635 to 1703, was the first to describe cells based upon his microscopic observations of cork. This illustration was published in his work Micrographia. Despite Hooke's early description of cells, their significance as the fundamental unit of life was not yet recognized. Nearly 200 years later, in 1838, Matthias Schleiden, 1804 to 1881, a German botanist who made extensive microscopic observations of plant tissues, described them as being composed of cells. Visualizing plant cells was relatively easy because plant cells are clearly separated by their thick cell walls. Schleiden believed that cells formed through crystallization rather than cell division. Theodor Schwann, 1810 to 1882, a noted German physiologist, made similar microscopic observations of animal tissue. In 1839, after a conversation with Schleiden, Schwann realized that similarities existed between plant and animal tissues. This laid the foundation for the idea that cells are the fundamental components of plants and animals. In the 1850s, 2 Polish scientists living in Germany pushed this idea further, culminating in what we recognize today as the modern cell theory. In 1852, Robert Remak, 1815 to 1865, a prominent neurologist and embryologist, published convincing evidence that cells are derived from other cells as a result of cell division. However, this idea was questioned by many in the scientific community. 3 years later, Rudolf Virchow, 1821 to 1902, a well-respected pathologist, published an editorial essay entitled Cellular Pathology, which popularized the concept of cell theory using the Latin phrase omnis cellula, a cellula. All cells arise from cells, which is essentially the second tenet of modern cell theory. Point 5. Given the similarity of Virchow's work to Remak's, there is some controversy as to which scientists should receive credit for articulating cell theory. See the following Ion Ethics feature for more about this controversy. Ion Ethics. Science and Plagiarism. Rudolf Virchow, a prominent Polish-born German scientist, is often remembered as the, quote, father of pathology. Well known for innovative approaches, he was one of the first to determine the causes of various diseases by examining their effects on tissues and organs. He was also among the first to use animals in his research, and, as a result of his work, he was the first to name numerous diseases and created many other medical terms. Over the course of his career, he published more than 2,000 papers and headed various important medical facilities, including the Charité Universitätsmedizin Berlin, a prominent Berlin hospital and medical school. But he is perhaps best remembered for his 1855 editorial essay titled Cellular Pathology, published in Archiv für Pathologische Anatomie und Physiologie, a journal that Virchow himself co-founded and still exists today. Despite his significant scientific legacy, there is some controversy regarding this essay, in which Virchow proposed the central tenet of modern cell theory, that all cells arise from other cells. Robert Remak, a former colleague who worked in the same laboratory as Virchow at the University of Berlin, had published the same idea 3 years before. Though it appears Virchow was familiar with Remak's work, he neglected to credit Remak's ideas in his essay. When Remak wrote a letter to Virchow pointing out similarities between Virchow's ideas and his own, Virchow was dismissive. In 1858, in the preface to one of his books, Virchow wrote that his 1855 publication was just an editorial piece, not a scientific paper, and thus there was no need to cite Remak's work. By today's standards, Virchow's editorial piece would certainly be considered an act of plagiarism, since he presented Remak's ideas as his own. However, in the 19th century, standards for academic integrity were much less clear. Virchow's strong reputation, coupled with the fact that Remak was a Jew, in a somewhat anti-Semitic political climate, shielded him from any significant repercussions. Today, the process of peer review and the ease of access to the scientific literature help discourage plagiarism. Although scientists are still motivated to publish original ideas that advance scientific knowledge, those who would consider plagiarizing are well aware of the serious consequences. In academia, plagiarism represents the theft of both individual thought and research, an offense that can destroy reputations and end careers. Figure 3.6a. Rudolf Virchow, 1821 to 1902, popularized the cell theory in an 1855 essay entitled Cellular Pathology. B. The idea that all cells originate from other cells was first published in 1852 by his contemporary and former colleague Robert Remak, 1815 to 1865. Check your understanding. What are the key points of the cell theory? What contributions did Rudolf Virchow and Robert Remak make to the development of the cell theory? Endosymbiotic theory. As scientists were making progress toward understanding the role of cells in plant and animal tissues, others were examining the structures within the cells themselves. In 1831, Scottish botanist Robert Brown, 1773 to 1858, was the first to describe observations of nuclei which he observed in plant cells. Then, in the early 1880s, German botanist Andreas Schimper, 1856 to 1901, was the first to describe the chloroplasts of plant cells, identifying their role in starch formation during photosynthesis and noting that they divided independent of the nucleus. Based upon the chloroplast's ability to reproduce independently, Russian botanist Konstantin Merezhkovsky, 1855 to 1921, suggested in 1905 that chloroplasts may have originated from ancestral photosynthetic bacteria living symbiotically inside a eukaryotic cell. He proposed a similar origin for the nucleus of plant cells. This was the first articulation of the endosymbiotic hypothesis and would explain how eukaryotic cells evolved from ancestral bacteria. Merezhkovsky's endosymbiotic hypothesis was furthered by American anatomist Ivan Wallin, 1883 to 1969, who began to experimentally examine the similarities between mitochondria, chloroplasts, and bacteria. In other words, to put the endosymbiotic hypothesis to the test using objective investigation. Wallin published a series of papers in the 1920s supporting the endosymbiotic hypothesis, including a 1926 publication co-authored with Merezhkovsky. Wallin claimed he could culture mitochondria outside of their eukaryotic host cells. Many scientists dismissed his cultures of mitochondria as resulting from bacterial contamination. Modern genome sequencing work supports the dissenting scientists by showing that much of the genome of mitochondria had been transferred to the host cell's nucleus, preventing the mitochondria from being able to live on their own. Wallen's ideas regarding the endosymbiotic hypothesis were largely ignored for the next 50 years because scientists were unaware that these organelles contained their own DNA. However, with the discovery of mitochondrial and chloroplast DNA in the 1960s, the endosymbiotic hypothesis was resurrected. Lynn Margulis, 1938–2011, an American geneticist, published her ideas regarding the endosymbiotic hypothesis of the origins of mitochondria and chloroplasts in 1967.12. In the decade leading up to her publication, advances in microscopy had allowed scientists to differentiate prokaryotic cells from eukaryotic cells. In her publication, Margulis reviewed the literature and argued that the eukaryotic organelles such as mitochondria and chloroplasts are of prokaryotic origin. She presented a growing body of microscopic, genetic, molecular biology, fossil, and geological data to support her claims. Again, this hypothesis was not initially popular, but mounting genetic evidence due to the advent of DNA sequencing supported the endosymbiotic theory, which is now defined as the theory that mitochondria and chloroplasts arose as a result of prokaryotic cells establishing a symbiotic relationship within a eukaryotic host, Figure 3.7. With Margulis's initial endosymbiotic theory gaining wide acceptance, she expanded on the theory in her 1981 book, Symbiosis in Cell Evolution. In it, she explains how endosymbiosis is a major driving factor in the evolution of organisms. More recent genetic sequencing and phylogenetic analysis show that mitochondrial DNA and chloroplast DNA are highly related to their bacterial counterparts, both in DNA sequence and chromosome structure. However, mitochondrial DNA and chloroplast DNA are reduced compared with nuclear DNA because many of the genes have moved from the organelles into the host cell's nucleus. Additionally, mitochondrial and chloroplast ribosomes are structurally similar to bacterial ribosomes, rather than to the eukaryotic ribosomes of their hosts. Last, the binary fission of these organelles strongly resembles the binary fission of bacteria, as compared with mitosis performed by eukaryotic cells. Since Margulis's original proposal, scientists have observed several examples of bacterial endosymbionts in modern-day eukaryotic cells. Examples include the endosymbiotic bacteria found within the guts of certain insects, such as cockroaches, 13, and photosynthetic bacteria-like organelles found in protists. Figure 3.7. According to the endosymbiotic theory, mitochondria and chloroplasts are each derived from the uptake of bacteria. These bacteria established a symbiotic relationship with their host cell that eventually led to the bacteria evolving into mitochondria and chloroplasts. Check your understanding. What does the modern endosymbiotic theory state? Main core. What evidence supports the endosymbiotic theory? Binai Shalalas. The germ theory of disease. Prior to the discovery of microbes during the 17th century, other theories circulated about the origins of disease. For example, the ancient Greeks proposed the miasma theory, which held that disease originated from particles emanating from decomposing matter, such as that in sewage or cesspits. Such particles infected humans in close proximity to the rotting material. Diseases including the Black Death, which ravaged Europe's population during the Middle Ages, were thought to have originated in this way. In the 11th century, Persian physician Ibn Sina, sometimes referred to as Avicenna, proposed that tuberculosis was likely spread by people's breath when in close proximity. Arab physician Ibn Zur, writing in about 1155, documented that the common skin condition scabies was caused by tiny mites that bored into the skin. Those scabies mites are about 0.5 millimeter long and therefore not technically microscopic, and their skin tunnels are often visible. Ibn Zur's discovery gave more evidence that unseen substances or creatures caused diseases. In 1546, Italian physician Girolamo Fracastoro proposed, in his essay De Contagione et Contagiosis Morbis, that seed-like spores may be transferred between individuals through direct contact, exposure to contaminated clothing, or through the air. We now recognize Fracastoro as an early proponent of the germ theory of disease, which states that diseases may result from microbial infection. However, in the 16th century, Fracastoro's ideas were not widely accepted and would be largely forgotten until the 19th century. In 1847, Hungarian obstetrician Ignaz Semmelweis, figure 3.8, observed that people who gave birth in hospital wards staffed by physicians and medical students were more likely to suffer and die from puerperal fever after childbirth, 10% to 20% mortality rate, than were people in wards staffed by midwives, 1% mortality rate. Semmelweis observed medical students performing autopsies and then subsequently carrying out vaginal examinations on living patients without washing their hands in between. He suspected that the students carried disease from the autopsies to the patients they examined. His suspicions were supported by the untimely death of a friend, a physician who contracted a fatal wound infection after a post-mortem examination of a woman who had died of a puerperal infection. The dead physician's wound had been caused by a scalpel used during the examination, and his subsequent illness and death closely paralleled that of the dead patient. Although Semmelweis did not know the true cause of puerperal fever, he proposed that physicians were somehow transferring the causative agent to their patients. He suggested that the number of puerperal fever cases could be reduced if physicians and medical students simply washed their hands with chlorinated lime water before and after examining every patient. When this practice was implemented, the maternal mortality rate in people cared for by physicians dropped to the same 1% mortality rate observed among people cared for by midwives. This demonstrated that hand washing was a very effective method for preventing disease transmission. Despite this great success, many discounted Semmelweis's work at the time, and physicians were slow to adopt the simple procedure of hand washing to prevent infections in their patients because it contradicted established norms for that time period. Figure 3.8 Ignaz Semmelweis, 1818 to 1865, was a proponent of the importance of hand washing to prevent transfer of disease between patients by physicians. Around the same time Semmelweis was promoting hand washing, in 1848, British physician John Snow conducted studies to track the source of cholera outbreaks in London. By tracing the outbreaks to two specific water sources, both of which were contaminated by sewage, Snow ultimately demonstrated that cholera bacteria were transmitted via drinking water. Snow's work is influential in that it represents the first known epidemiological study, and it resulted in the first known public health response to an epidemic. The work of both Semmelweis and Snow clearly refuted the prevailing miasma theory of the day, showing that disease is not only transmitted through the air, but also through contaminated items. Although the work of Semmelweis and Snow successfully showed the role of sanitation in preventing infectious disease, the cause of disease was not fully understood. The subsequent work of Louis Pasteur, Robert Koch, and Joseph Lister would further substantiate the germ theory of disease. While studying the causes of beer and wine spoilage in 1856, Pasteur discovered properties of fermentation by microorganisms. He had demonstrated with his swan neck flask experiments, figure 3.4, that airborne microbes, not spontaneous generation, were the cause of food spoilage, and he suggested that if microbes were responsible for food spoilage and fermentation, they could also be responsible for causing infection. This was the foundation for the germ theory of disease. Meanwhile, British surgeon Joseph Lister, figure 3.9, was trying to determine the causes of post-surgical infections. Many physicians did not give credence to the idea that microbes on their hands, on their clothes, or in the air could infect patients' surgical wounds, despite the fact that 50% of surgical patients, on average, were dying of post-surgical infections. 0.15. Lister, however, was familiar with the work of Semmelweis and Pasteur, therefore, he insisted on hand washing and extreme cleanliness during surgery. In 1867, to further decrease the incidence of post-surgical wound infections, Lister began using carbolic acid phenol, spray disinfectant/antiseptic during surgery. His extremely successful efforts to reduce post-surgical infection caused his techniques to become a standard medical practice. A few years later, Robert Koch, figure 3.9, proposed a series of postulates, Koch's postulates, based on the idea that the cause of a specific disease could be attributed to a specific microbe. Using these postulates, Koch and his colleagues were able to definitively identify the causative pathogens of specific diseases, including anthrax, tuberculosis, and cholera. Koch's one microbe, one disease concept was the culmination of the 19th century's paradigm shift away from miasma theory and toward the germ theory of disease. Koch's postulates are discussed more thoroughly in How Pathogens Cause Disease. Figure 3.9a, Joseph Lister developed procedures for the proper care of surgical wounds and the sterilization of surgical equipment. B. Robert Koch established a protocol to determine the cause of infectious disease. Both scientists contributed significantly to the acceptance of the germ theory of disease. Check your understanding. Compare and contrast the miasma theory of disease with the germ theory of disease. How did Joseph Lister's work contribute to the debate between the miasma theory and germ theory, and how did this increase the success of medical procedures? Clinical focus. Part 2. After suffering a fever, congestion, cough, and increasing aches and pains for several days, Barbara suspects that she has a case of the flu. She decides to visit the health center at her university. The PA tells Barbara that her symptoms could be due to a range of diseases, such as influenza, bronchitis, pneumonia, or tuberculosis. During her physical examination, the PA notes that Barbara's heart rate is slightly elevated. Using a pulse oximeter, a small device that clips on her finger, he finds that Barbara has hypoxemia, a lower than normal level of oxygen in the blood. Using a stethoscope, the PA listens for abnormal sounds made by Barbara's heart, lungs, and digestive system. As Barbara breathes, the PA hears a crackling sound and notes a slight shortness of breath. He collects a sputum sample, noting the greenish color of the mucus, and orders a chest radiograph, which shows a shadow in the left lung. All of these signs are suggestive of pneumonia, a condition in which the lungs fill with mucus. Figure 3.10. Figure 3.10. This is a chest radiograph typical of pneumonia. Because X-ray images are negative images, a shadow is seen as a white area within the lung that should otherwise be black. In this case, the left lung shows a shadow as a result of pockets in the lung that have become filled with fluid. Credit left. Modification of work by Christaris A. Wikimedia Commons. What kinds of infectious agents are known to cause pneumonia? Jump to the next clinical focus box. Go back to the previous clinical focus box. Figure 3.11. Credit Swan Neck Flask. Modification of work by Wellcome Images. Learning objectives. By the end of this section, you will be able to explain the distinguishing characteristics of prokaryotic cells, describe common cell morphologies and cellular arrangements typical of prokaryotic cells, and explain how cells maintain their morphology. Describe internal and external structures of prokaryotic cells in terms of their physical structure, chemical structure, and function. Compare the distinguishing characteristics of bacterial and archaeal cells. Cell theory states that the cell is the fundamental unit of life. However, cells vary significantly in size, shape, structure, and function. At the simplest level of construction, all cells possess a few fundamental components. These include cytoplasm, a gel-like substance composed of water and dissolved chemicals needed for growth, which is contained within a plasma membrane, also called a cell membrane or cytoplasmic membrane. One or more chromosomes, which contain the genetic blueprints of the cell, and ribosomes, organelles used for the production of proteins. Beyond these basic components, cells can vary greatly between organisms and even within the same multicellular organism. The two largest categories of cells, prokaryotic cells and eukaryotic cells, are defined by major differences in several cell structures. Prokaryotic cells lack a nucleus surrounded by a complex nuclear membrane and generally have a single, circular chromosome located in a nucleoid. Eukaryotic cells have a nucleus surrounded by a complex nuclear membrane that contains multiple, rod-shaped chromosomes. 0.16. All plant cells and animal cells are eukaryotic. Some microorganisms are composed of prokaryotic cells, whereas others are composed of eukaryotic cells. Prokaryotic microorganisms are classified within the domains archaea and bacteria, whereas eukaryotic organisms are classified within the domain eukarya. The structures inside a cell are analogous to the organs inside a human body, with unique structures suited to specific functions. Some of the structures found in prokaryotic cells are similar to those found in some eukaryotic cells. Others are unique to prokaryotes. Although there are some exceptions, eukaryotic cells tend to be larger than prokaryotic cells. The comparatively larger size of eukaryotic cells dictates the need to compartmentalize various chemical processes within different areas of the cell, using complex membrane-bound organelles. In contrast, prokaryotic cells generally lack membrane-bound organelles. However, they often contain inclusions that compartmentalize their cytoplasm. Figure 3.12 illustrates structures typically associated with prokaryotic cells. These structures are described in more detail in the next section. Figure 3.12. A typical prokaryotic cell contains a cell membrane, chromosomal DNA that is concentrated in a nucleoid, ribosomes, and a cell wall. Some prokaryotic cells may also possess flagella, pili, fimbriae, and capsules, common cell morphologies and arrangements. Individual cells of a particular prokaryotic organism are typically similar in shape or cell morphology. Although thousands of prokaryotic organisms have been identified, only a handful of cell morphologies are commonly seen microscopically. Figure 3.13 names and illustrates cell morphologies commonly found in prokaryotic cells. In addition to cellular shape, prokaryotic cells of the same species may group together in certain distinctive arrangements depending on the plane of cell division. Some common arrangements are shown in Figure 3.14. Common prokaryotic cell shapes. The term coccus, plural cocci, is the name given to round, spherical shapes. The term bacillus, plural bacilli, is the name given to rod-shaped cells. These cells are shaped like long, rounded rectangles. The term vibrio, plural vibrios, is the name given to curved rods. These cells have a shape like a long comma. The term coccobacillus, plural coccobacilli, is the name for short rods. These cells look like ovals. The term spirillum, plural spirilla, is the name for long spiral cells. These look like corkscrews. The term spirochete, plural spirochetes, is the name for long, loose helical spiral-shaped cells. These look similar to the spirillum, but are more floppy. Figure 3.13. Credit Coccus Micrograph. Modification of work by Janice Haney-Carr, Centers for Disease Control and Prevention. Credit Coccobacillus Micrograph. Modification of work by Janice Carr, Centers for Disease Control and Prevention. Credit Spirochete Micrograph. Modification of work by Centers for Disease Control and Prevention. Figure 3.14. In most prokaryotic cells, morphology is maintained by the cell wall in combination with cytoskeletal elements. The cell wall is a structure found in most prokaryotes and some eukaryotes. It envelopes the cell membrane, protecting the cell from changes in osmotic pressure. Figure 3.15. Osmotic pressure occurs because of differences in the concentration of solutes on opposing sides of a semipermeable membrane. Water is able to pass through a semipermeable membrane, but solutes, dissolved molecules like salts, sugars, and other compounds, cannot. When the concentration of solutes is greater on one side of the membrane, water diffuses across the membrane from the side with the lower concentration, more water, to the side with the higher concentration, less water, until the concentrations on both sides become equal. This diffusion of water is called osmosis, and it can cause extreme osmotic pressure on a cell when its external environment changes. The external environment of a cell can be described as an isotonic, hypertonic, or hypotonic medium. In an isotonic medium, the solute concentrations inside and outside the cell are approximately equal, so there is no net movement of water across the cell membrane. In a hypertonic medium, the solute concentration outside the cell exceeds that inside the cell, so water diffuses out of the cell and into the external medium. In a hypotonic medium, the solute concentration inside the cell exceeds that outside of the cell, so water will move by osmosis into the cell. This causes the cell to swell and potentially lyse or burst. The degree to which a particular cell is able to withstand changes in osmotic pressure is called tonicity. Cells that have a cell wall are better able to withstand subtle changes in osmotic pressure and maintain their shape. In hypertonic environments, cells that lack a cell wall can become dehydrated, causing crenation or shriveling of the cell. The plasma membrane contracts and appears scalloped or notched, Figure 3.15. By contrast, cells that possess a cell wall undergo plasmolysis rather than crenation. In plasmolysis, the plasma membrane contracts and detaches from the cell wall, and there is a decrease in interior volume, but the cell wall remains intact, thus allowing the cell to maintain some shape and integrity for a period of time. Figure 3.16. Likewise, cells that lack a cell wall are more prone to lysis in hypotonic environments. The presence of a cell wall allows the cell to maintain its shape and integrity for a longer time before lysing. Figure 3.16. Figure 3.15. In cells that lack a cell wall, changes in osmotic pressure can lead to crenation in hypertonic environments or cell lysis in hypotonic environments. Figure 3.16. In prokaryotic cells, the cell wall provides some protection against changes in osmotic pressure, allowing it to maintain its shape longer. The cell membrane is typically attached to the cell wall in an isotonic medium left. In a hypertonic medium, the cell membrane detaches from the cell wall and contracts, plasmolysis, as water leaves the cell. In a hypotonic medium right, the cell wall prevents the cell membrane from expanding to the point of bursting, although lysis will eventually occur if too much water is absorbed. Check your understanding. Explain the difference between cell morphology and arrangement. What advantages do cell walls provide prokaryotic cells? The nucleoid. All cellular life has a DNA genome organized into one or more chromosomes. Prokaryotic chromosomes are typically circular, haploid unpaired, and not bound by a complex nuclear membrane. Prokaryotic DNA and DNA-associated proteins are concentrated within the nucleoid region of the cell, figure 3.17. In general, prokaryotic DNA interacts with nucleoid-associated proteins, NAPs, that assist in the organization and packaging of the chromosome. In bacteria, NAPs function similar to histones, which are the DNA-organizing proteins found in eukaryotic cells. In archaea, the nucleoid is organized by either NAPs or histone-like DNA-organizing proteins. Figure 3.17. The nucleoid region, the area enclosed by the green dashed line, is a condensed area of DNA found within prokaryotic cells. Because of the density of the area, it does not readily stain and appears lighter in color when viewed with a transmission electron microscope. Plasmids. Prokaryotic cells may also contain extrachromosomal DNA, or DNA, that is not part of the chromosome. This extrachromosomal DNA is found in plasmids, which are small, circular, double-stranded DNA molecules. Cells that have plasmids often have hundreds of them within a single cell. Plasmids are more commonly found in bacteria. However, plasmids have been found in archaea and eukaryotic organisms. Plasmids often carry genes that confer advantageous traits, such as antibiotic resistance. Thus, they are important to the survival of the organism. We will discuss plasmids in more detail in mechanisms of microbial genetics. Ribosomes. All cellular life synthesizes proteins, and organisms in all three domains of life possess ribosomes, structures responsible for protein synthesis. However, ribosomes in each of the three domains are structurally different. Ribosomes themselves are constructed from proteins, along with ribosomal RNA, rRNA. Prokaryotic ribosomes are found in the cytoplasm. They're called 70S ribosomes because they have a size of 70S, figure 3.18, whereas eukaryotic cytoplasmic ribosomes have a size of 80S. The S stands for Svedberg unit, a measure of sedimentation in an ultracentrifuge, which is based on size, shape, and surface qualities of the structure being analyzed. Although they are the same size, bacterial and archaeal ribosomes have different proteins and rRNA molecules, and the archaeal versions are more similar to their eukaryotic counterparts than to those found in bacteria. Figure 3.18. Prokaryotic ribosomes, 70S, are composed of two subunits, the 30S, small subunit, and the 50S, large subunit, each of which are composed of protein and rRNA components. Inclusions. As single-celled organisms living in unstable environments, some prokaryotic cells have the ability to store excess nutrients within cytoplasmic structures, called inclusions. Storing nutrients in a polymerized form is advantageous because it reduces the buildup of osmotic pressure that occurs as a cell accumulates solutes. Various types of inclusions store glycogen and starches, which contain carbon that cells can access for energy. Volutin granules, also called metachromatic granules because of their staining characteristics, are inclusions that store polymerized inorganic phosphate that can be used in metabolism and assist in the formation of biofilms. Microbes known to contain volutin granules include the Archaea methanosarcina, the bacterium Corynebacterium diphtheriae, and the unicellular eukaryotic alga Chlamydomonas. Sulfur granules, another type of inclusion, are found in sulfur bacteria of the genus Thiobacillus. These granules store elemental sulfur, which the bacteria use for metabolism. Occasionally, certain types of inclusions are surrounded by a phospholipid monolayer embedded with protein. Polyhydroxybutyrate, PHB, which can be produced by species of Bacillus and Pseudomonas, is an example of an inclusion that displays this type of monolayer structure. Industrially, PHB has also been used as a source of biodegradable polymers for bioplastics. Several different types of inclusions are shown in Figure 3.19. Figure 3.19. Prokaryotic cells may have various types of inclusions. A transmission electron micrograph of polyhydroxybutyrate lipid droplets. B. A light micrograph of volutin granules. C. A phase contrast micrograph of sulfur granules. D. A transmission electron micrograph of gas vacuoles. E. A transmission electron micrograph of magnetosomes. Credit B, C, D. Modification of work by American Society for Microbiology. Some prokaryotic cells have other types of inclusions that serve purposes other than nutrient storage. For example, some prokaryotic cells produce gas vacuoles, accumulations of small protein-lined vesicles of gas. These gas vacuoles allow the prokaryotic cells that synthesize them to alter their buoyancy so that they can adjust their location in the water column. Magnetotactic bacteria, such as Magnetospirillum magnetotacticum, contain magnetosomes, which are inclusions of magnetic iron oxide or iron sulfide surrounded by a lipid layer. These allow cells to align along a magnetic field, aiding their movement. Figure 3.19. Cyanobacteria such as Anabaena cylindrica and bacteria such as Halothiobacillus neapolitanus produce carboxysome inclusions. Carboxysomes are composed of outer shells of thousands of protein subunits. Their interior is filled with ribulose 1,5-bisphosphate carboxylase/oxygenase, rubisco, and carbonic anhydrase. Both of these compounds are used for carbon metabolism. Some prokaryotic cells also possess carboxysomes that sequester functionally related enzymes in one location. These structures are considered proto-organelles because they compartmentalize important compounds or chemical reactions, much like many eukaryotic organelles. Endospores. Bacterial cells are generally observed as vegetative cells, but some genera of bacteria have the ability to form endospores, structures that essentially protect the bacterial genome in a dormant state when environmental conditions are unfavorable. Endospores, not to be confused with the reproductive spores formed by fungi, allow some bacterial cells to survive long periods without food or water, as well as exposure to chemicals, extreme temperatures, and even radiation. Table 3.1 compares the characteristics of vegetative cells and endospores. Characteristics of vegetative cells versus endospores. Vegetative cells, endospores. Sensitive to extreme temperatures and radiation. Resistant to extreme temperatures and radiation. Nearly all endospore-producing bacteria gram-positive, as vegetative cells do not absorb gram stain, only special endospore stains. See staining microscopic specimens. Normal water content and enzymatic activity. Dehydrated, no metabolic activity. Capable of active growth and metabolism. Dormant, no growth or metabolic activity. Table 3.1. The process by which vegetative cells transform into endospores is called sporulation, and it generally begins when nutrients become depleted or environmental conditions become otherwise unfavorable. Figure 3.20. The process begins with the formation of a septum in the vegetative bacterial cell. The septum divides the cell asymmetrically, separating a DNA forespore from the mother cell. The forespore, which will form the core of the endospore, is essentially a copy of the cell's chromosomes and is separated from the mother cell by a second membrane. A cortex gradually forms around the forespore by laying down layers of calcium and dipicolinic acid between membranes. A protein coat then forms around the cortex while the DNA of the mother cell disintegrates. Further maturation of the endospore occurs with the formation of an outermost exosporium. The endospore is released upon disintegration of the mother cell, completing sporulation. Figure 3.20, asporulation begins following asymmetric cell division. The forespore becomes surrounded by a double layer of membrane, a cortex, and a protein coat, before being released as a mature endospore upon disintegration of the mother cell. B. An electron micrograph of a carboxydathermus hydrogenoformans endospore. C. These Bacillus spp cells are undergoing sporulation. The endospores have been visualized using malachite green stain. Credit B. Modification of work by Jonathan Eisen. Endospores of certain species have been shown to persist in a dormant state for extended periods of time, up to thousands of years. 17. However, when living conditions improve, endospores undergo germination, re-entering a vegetative state. After germination, the cell becomes metabolically active again and is able to carry out all of its normal functions, including growth and cell division. Not all bacteria have the ability to form endospores. However, there are a number of clinically significant endospore-forming gram-positive bacteria of the genera Bacillus and Clostridium. These include B. anthracis, the causative agent of anthrax, which produces endospores capable of surviving for many decades. 18. C. tetani causes tetanus. C. perfringens causes gas gangrene. And C. botulinum causes botulism. Clostridioides difficile is a distant relative causing pseudomembranous colitis. Pathogens such as these are particularly difficult to combat because their endospores are so hard to kill. Special sterilization methods for endospore-forming bacteria are discussed in control of microbial growth. Check your understanding. What is an inclusion? Alome. What is the function of an endospore? Alome. Plasma membrane. Structures that enclose the cytoplasm and internal structures of the cell are known collectively as the cell envelope. In prokaryotic cells, the structures of the cell envelope vary depending on the type of cell and organism. Most, but not all, prokaryotic cells have a cell wall, but the makeup of this cell wall varies. All cells, prokaryotic and eukaryotic, have a plasma membrane, also called cytoplasmic membrane or cell membrane, that exhibits selective permeability, allowing some molecules to enter or leave the cell while restricting the passage of others. The structure of the plasma membrane is often described in terms of the fluid mosaic model, which refers to the ability of membrane components to move fluidly within the plane of the membrane, as well as the mosaic-like composition of the components, which include a diverse array of lipid and protein components. Figure 3.21. The plasma membrane structure of most bacterial and eukaryotic cell types is a bilayer composed mainly of phospholipids, formed with ester linkages and proteins. These phospholipids and proteins have the ability to move laterally within the plane of the membranes as well as between the two phospholipid layers. Figure 3.21. The bacterial plasma membrane is a phospholipid bilayer with a variety of embedded proteins that perform various functions for the cell. Note the presence of glycoproteins and glycolipids, whose carbohydrate components extend out from the surface of the cell. The abundance and arrangement of these proteins and lipids can vary greatly between species. Archaeal membranes are fundamentally different from bacterial and eukaryotic membranes in a few significant ways. First, archaeal membrane phospholipids are formed with ether linkages in contrast to the ester linkages found in bacterial or eukaryotic cell membranes. Second, archaeal phospholipids have branched chains, whereas those of bacterial and eukaryotic cells are straight-chained. Finally, although some archaeal membranes can be formed of bilayers like those found in bacteria and eukaryotes, other archaeal plasma membranes are lipid monolayers. Proteins on the cell surface are important for a variety of functions, including cell-to-cell communication and sensing environmental conditions and pathogenic virulence factors. Membrane proteins and phospholipids may have carbohydrates, sugars associated with them and are called glycoproteins or glycolipids, respectively. These glycoprotein and glycolipid complexes extend out from the surface of the cell, allowing the cell to interact with the external environment. Figure 3.21. Glycoproteins and glycolipids in the plasma membrane can vary considerably in chemical composition among archaea, bacteria, and eukaryotes, allowing scientists to use them to characterize unique species. Plasma membranes from different cell types also contain unique phospholipids, which contain fatty acids. As described in using biochemistry to identify microorganisms, phospholipid-derived fatty acid analysis (PLFA) profiles can be used to identify unique types of cells based on differences in fatty acids. Archaea, bacteria, and eukaryotes each have a unique PFLA profile, membrane transport mechanisms. One of the most important functions of the plasma membrane is to control the transport of molecules into and out of the cell. Internal conditions must be maintained within a certain range despite any changes in the external environment. The transport of substances across the plasma membrane allows cells to do so. Cells use various modes of transport across the plasma membrane. For example, molecules moving from a higher concentration to a lower concentration with the concentration gradient are transported by simple diffusion, also known as passive transport. Figure 3.22. Some small molecules, like carbon dioxide, may cross the membrane bilayer directly by simple diffusion. However, charged molecules, as well as large molecules, need the help of carriers or channels in the membrane. These structures ferry molecules across the membrane, a process known as facilitated diffusion, figure 3.23. Active transport occurs when cells move molecules across their membrane against concentration gradients, figure 3.24. A major difference between passive and active transport is that active transport requires adenosine triphosphate, ATP, or other forms of energy to move molecules uphill. Therefore, active transport structures are often called pumps, figure 3.22. Simple diffusion down a concentration gradient directly across the phospholipid bilayer. Credit: Modification of work by Mariana Ruiz Villarreal. Figure 3.23. Facilitated diffusion down a concentration gradient through a membrane protein. Credit: Modification of work by Mariana Ruiz Villarreal. Figure 3.24. Active transport against a concentration gradient via a membrane pump that requires energy. Credit: Modification of work by Mariana Ruiz Villarreal. Group translocation also transports substances into bacterial cells. In this case, as a molecule moves into a cell against its concentration gradient, it is chemically modified so that it does not require transport against an unfavorable concentration gradient. A common example of this is the bacterial phosphotransferase system, a series of carriers that phosphorylates, i.e., adds phosphate ions to glucose or other sugars upon entry into cells. Since the phosphorylation of sugars is required during the early stages of sugar metabolism, the phosphotransferase system is considered to be an energy-neutral system. Photosynthetic membrane structures. Some prokaryotic cells, namely cyanobacteria and photosynthetic bacteria, have membrane structures that enable them to perform photosynthesis. These structures consist of an unfolding of the plasma membrane that encloses photosynthetic pigments, such as green chlorophylls and bacteriochlorophylls. In cyanobacteria, these membrane structures are called thylakoids. In photosynthetic bacteria, they are called chromatophores, lamella, or chlorosomes, cell wall. The primary function of the cell wall is to protect the cell from harsh conditions in the outside environment. When present, there are notable similarities and differences among the cell walls of archaea, bacteria, and eukaryotes. The major component of bacterial cell walls is called peptidoglycan, or muran. It is only found in bacteria. Structurally, peptidoglycan resembles a layer of meshwork or fabric, figure 3.25. Each layer is composed of long chains of alternating molecules of N-acetylglucosamine, NAG, and N-acetylmuramic acid, NAM. The structure of the long chains has significant two-dimensional tensile strength due to the formation of peptide bridges that connect NAG, N-NAG, and NAM within each peptidoglycan layer. In gram-negative bacteria, tetrapeptide chains extending from each NAM unit are directly cross-linked, whereas in gram-positive bacteria, these tetrapeptide chains are linked by pentaglycine cross bridges. Peptidoglycan subunits are made inside of the bacterial cell and then exported and assembled in layers, giving the cell its shape. Since peptidoglycan is unique to bacteria, many antibiotic drugs are designed to interfere with peptidoglycan synthesis, weakening the cell wall and making bacterial cells more susceptible to the effects of osmotic pressure. See Mechanisms of antibacterial drugs. In addition, Certain cells of the human immune system are able to recognize bacterial pathogens by detecting peptidoglycan on the surface of a bacterial cell. These cells then engulf and destroy the bacterial cell using enzymes such as lysozyme, which breaks down and digests the peptidoglycan in their cell walls. See pathogen recognition and phagocytosis. Figure 3.25 peptidoglycan is composed of polymers of alternating NAM and NAG subunits, which are cross-linked by peptide bridges linking NAM subunits from various glycan chains. This provides the cell wall with tensile strength in 2 dimensions. The Gram staining protocol, see staining microscopic specimens, is used to differentiate 2 common types of cell wall structures. Figure 3.26. Gram-positive cells have a cell wall consisting of many layers of peptidoglycan totaling 30 to 100 nanometers in thickness. These peptidoglycan layers are commonly embedded with teichoic acids, TAs, carbohydrate chains that extend through and beyond the peptidoglycan layer. 0.19 TA is thought to stabilize peptidoglycan by increasing its rigidity. TA also plays a role in the ability of pathogenic gram-positive bacteria, such as Streptococcus, to bind to certain proteins on the surface of host cells, enhancing their ability to cause infection. In addition to peptidoglycan and TAs, bacteria of the family Mycobacteriaceae have an external layer of waxy mycolic acids in their cell wall. As described in staining microscopic specimens, these bacteria are referred to as acid-fast, since acid-fast stains must be used to penetrate the mycolic acid layer for purposes of microscopy. Figure 3.27. Figure 3.26 bacteria contain 2 common cell wall structural types. Gram-positive cell walls are structurally simple, containing a thick layer of peptidoglycan with embedded teichoic acid external to the plasma membrane. Gram-negative cell walls are structurally more complex, containing a thin layer of peptidoglycan and an outer membrane containing lipopolysaccharide. Credit: Modification of work by Francis Koob, 2 inches /Wikimedia Commons. Figure 3.27a. Some gram-positive bacteria, including members of the Mycobacteriaceae, produce waxy mycolic acids found exterior to their structurally distinct peptidoglycan. B. The acid-fast staining protocol detects the presence of cell walls that are rich in mycolic acid. Acid-fast cells are stained red by carbylfusion. Credit A. Modification of work by Francis Posp, 2 inches Wikimedia Commons. Credit B. Modification of work by Centers for Disease Control and Prevention. Gram-negative cells have a much thinner layer of peptidoglycan, no more than about 4 nanometers thick 21, than gram-positive cells, and the overall structure of their cell envelope is more complex. In gram-negative cells, a gel-like matrix occupies the periplasmic space between the cell wall and the plasma membrane, and there is a second lipid bilayer called the outer membrane, which is external to the peptidoglycan layer. Figure 3.26. This outer membrane is attached to the peptidoglycan by murine lipoprotein. The outer leaflet of the outer membrane contains the molecule lipopolysaccharide, LPS, which functions as an endotoxin in infections involving gram-negative bacteria, contributing to symptoms such as fever, hemorrhaging, and septic shock. Each LPS molecule is composed of lipid A, a core polysaccharide, and an O-side chain that is composed of sugar-like molecules that comprise the external face of the LPS. Figure 3.28. The composition of the O-side chain varies between different species and strains of bacteria. Parts of the O-side chain called antigens can be detected using serological or immunological tests to identify specific pathogenic strains like Escherichia coli O157H7, a deadly strain of bacteria that causes bloody diarrhea and kidney failure. Figure 3.28. The outer membrane of a gram-negative bacterial cell contains lipopolysaccharide, LPS, a toxin composed of lipid A embedded in the outer membrane, a core polysaccharide, and the O-side chain. Archaeal cell wall structure differs from that of bacteria in several significant ways. First, archaeal cell walls do not contain peptidoglycan. Instead, they contain a similar polymer called pseudopeptidoglycan, pseudomurane, in which NAM is replaced with a different subunit. Other archaea may have a layer of glycoproteins or polysaccharides that serves as the cell wall instead of pseudopeptidoglycan. Last, as is the case with some bacterial species, there are a few archaea that appear to lack cell walls entirely: glycochalaces and S layers. Although most prokaryotic cells have cell walls, some may have additional cell envelope structures exterior to the cell wall, such as glycocalyx and S layers. A glycocalyx is a sugar coat, of which there are two important types: capsules and slime layers. A capsule is an organized layer located outside of the cell wall and usually composed of polysaccharides or proteins, figure 3.29. A slime layer is a less tightly organized layer that is only loosely attached to the cell wall and can be more easily washed off. Lime layers may be composed of polysaccharides, glycoproteins, or glycolipids. Glycocalyces allows cells to adhere to surfaces, aiding in the formation of biofilms, colonies of microbes that form in layers on surfaces. In nature, most microbes live in mixed communities within biofilms, partly because the biofilm affords them some level of protection. Biofilms generally hold water like a sponge, preventing desiccation. They also protect cells from predation and hinder the action of antibiotics and disinfectants. All of these properties are advantageous to the microbes living in a biofilm, but they present challenges in a clinical setting, where the goal is often to eliminate microbes. Figure 3.29a capsules are a type of glycocalyx composed of an organized layer of polysaccharides. B. A capsule stain of Pseudomonas aeruginosa, a bacterial pathogen capable of causing many different types of infections in humans. Credit B. Modification of work by American Society for Microbiology. The ability to produce a capsule can contribute to a microbe's pathogenicity, ability to cause disease, because the capsule can make it more difficult for phagocytic cells, such as white blood cells, to engulf and kill the microorganism. Streptococcus pneumoniae, for example, produces a capsule that is well known to aid in this bacterium's pathogenicity. As explained in staining microscopic specimens, capsules are difficult to stain for microscopy. Negative staining techniques are typically used. An S layer is another type of cell envelope structure. It is composed of a mixture of structural proteins and glycoproteins. In bacteria, S layers are found outside the cell wall, but in some archaea, the S layer serves as the cell wall. The exact function of S layers is not entirely understood, and they are difficult to study, but available evidence suggests that they may play a variety of functions in different prokaryotic cells, such as helping the cell withstand osmotic pressure and for certain pathogens interacting with the host immune system. Clinical focus. Part 3. After diagnosing Barbara with pneumonia, the PA writes her a prescription for amoxicillin, a commonly prescribed type of penicillin derivative. More than a week later, despite taking the full course as directed, Barbara still feels weak and is not fully recovered, although she is still able to get through her daily activities. She returns to the health center for a follow-up visit. Many types of bacteria, fungi, and viruses can cause pneumonia. Amoxicillin targets the peptidoglycan of bacterial cell walls. Since the amoxicillin has not resolved Barbara's symptoms, the PA concludes that the causative agent probably lacks peptidoglycan, meaning that the pathogen could be a virus, a fungus, or a bacterium that lacks peptidoglycan. Another possibility is that the pathogen is a bacterium containing peptidoglycan, but has developed resistance to amoxicillin. How can the PA definitively identify the cause of Barbara's pneumonia? What form of treatment should the PA prescribe, given that the amoxicillin was ineffective? Jump to the next clinical focus box. Go back to the previous clinical focus box. Filamentous appendages. Many bacterial cells have protein appendages embedded within their cell envelopes that extend outward, allowing interaction with the environment. These appendages can attach to other surfaces, transfer DNA, or provide movement. Filamentous appendages include fimbriae, pili, and flagella. Fimbriae and pili. Fimbriae and pili are structurally similar, and because differentiation between the two is problematic, these terms are often used interchangeably. 2223. The term fimbriae commonly refers to short bristle-like proteins projecting from the cell surface by the hundreds. Fimbriae enable a cell to attach to surfaces and to other cells. For pathogenic bacteria, adherence to host cells is important for colonization, infectivity, and virulence. Adherence to surfaces is also important in biofilm formation. The term pili, singular, pilus, commonly refers to longer, less numerous protein appendages that aid in attachment to surfaces. Figure 3.30. A specific type of pilus, called the F pilus or sex pilus, is important in the transfer of DNA between bacterial cells, which occurs between members of the same generation when two cells physically transfer or exchange parts of their respective genomes. See how asexual prokaryotes achieve genetic diversity. Figure 3.30. Bacteria may produce two different types of protein appendages that aid in surface attachment. Fimbriae typically are more numerous and shorter, whereas pili, shown here, are longer and less numerous per cell. Credit: Modification of work by American Society for Microbiology. Microconnections. Group A, strep. Before the structure and function of the various components of the bacterial cell envelope were well understood, scientists were already using cell envelope characteristics to classify bacteria. In 1933, Rebecca Lansfield proposed a method for serotyping various beta-hemolytic strains of Streptococcus species using an agglutination assay, a technique using the clumping of bacteria to detect specific cell surface antigens. In doing so, Lansfield discovered that one group of S. pyogenes, found in Group A, was associated with a variety of human diseases. She determined that various strains of Group A strep could be distinguished from each other based on variations in specific cell surface proteins that she named M proteins. Today, more than 80 different strains of Group A strep have been identified based on M proteins. Various strains of Group A strep are associated with a wide variety of human infections, including streptococcal pharyngitis, strep throat, impetigo, toxic shock syndrome, scarlet fever, rheumatic fever, and necrotizing fasciitis. The M protein is an important virulence factor for Group A strep, helping these strains evade the immune system. Changes in M proteins appear to alter the infectivity of a particular strain of Group A strep. Flagella. Flagella are structures used by cells to move in aqueous environments. Bacterial flagella act like propellers. They are stiff spiral filaments composed of flagellin protein subunits that extend outward from the cell and spin in solution. The basal body is the motor for the flagellum and is embedded in the plasma membrane. Figure 3.31. A hook region connects the basal body to the filament. Gram-positive and gram-negative bacteria have different basal body configurations due to differences in cell wall structure. Different types of motile bacteria exhibit different arrangements of flagella. Figure 3.32. A bacterium with a singular flagellum, typically located at one end of the cell, polar, is said to have a monotrichous flagellum. An example of a monotrichously flagellated bacterial pathogen is Vibrio cholerae, the gram-negative bacterium that causes cholera. Cells with amphitrichous flagella have a flagellum or tufts of flagella at each end. An example is Spirillum minor, the cause of spirillary Asian, ratbite fever, or Sudoku. Cells with Lophotrichous flagella have a tuft at one end of the cell. The gram-negative Bacillus Pseudomonas aeruginosa, an opportunistic pathogen known for causing many infections, including swimmer's ear and burn wound infections, has Lophotrichous flagella. Flagella that cover the entire surface of a bacterial cell are called peritrichous flagella. The gram-negative bacterium E. coli shows a peritrichous arrangement of flagella. Figure 3.31. The basic structure of a bacterial flagellum consists of a basal body, hook, and filament. The basal body composition and arrangement differ between gram-positive and gram-negative bacteria. Credit: Modification of work by Lady of Hats, Mariana Ruiz Villarreal. Figure 3.32. Flagellated bacteria may exhibit multiple arrangements of their flagella. Common arrangements include monotrichous, amphitrichous, lophotrichous, or peritrichous. Directional movement depends on the configuration of the flagella. Bacteria can move in response to a variety of environmental signals, including light, phototaxis, magnetic fields, magnetotaxis, using magnetosomes, and, most commonly, chemical gradients, chemotaxis. Purposeful movement toward a chemical attractant, like a food source, or away from a repellent, like a poisonous chemical, is achieved by increasing the length of runs and decreasing the length of tumbles. When running, flagella rotate in a counterclockwise direction, allowing the bacterial cell to move forward. In a Paratrichus bacterium, the flagella are all bundled together in a very streamlined way, figure 3.33, allowing for efficient movement. When tumbling, flagella are splayed out while rotating in a clockwise direction, creating a looping motion and preventing meaningful forward movement but reorienting the cell toward the direction of the attractant. When an attractant exists, runs and tumbles still occur. However, the length of runs is longer, while the length of the tumbles is reduced, allowing overall movement toward the higher concentration of the attractant. When no chemical gradient exists, the lengths of runs and tumbles are more equal, and overall movement is more random. Figure 3.34. Figure 3.33 bacteria achieve directional movement by changing the rotation of their flagella. In a cell with Paratrichus flagella, the flagella bundle when they rotate in a counterclockwise direction, resulting in a run. However, when the flagella rotate in a clockwise direction, the flagella are no longer bundled, resulting in tumbles. Figure 3.34. Without a chemical gradient, flagella rotation cycles between counterclockwise run and clockwise tumble, with no overall directional movement. However, when a chemical gradient of an attractant exists, the length of runs is extended, while the length of tumbles is decreased. This leads to chemotaxis, an overall directional movement toward the higher concentration of the attractant. Check your understanding. What is the peptidoglycan layer, and how does it differ between gram-positive and gram-negative bacteria? Compare and contrast monotrichous, amphitrichous, lophotrichous, and paratrichous flagella. Learning objectives. By the end of this section, you will be able to explain the distinguishing characteristics of eukaryotic cells, describe internal and external structures of eukaryotic cells in terms of their physical structure, chemical structure, and function. Identify and describe structures and organelles unique to eukaryotic cells. Compare and contrast similar structures found in prokaryotic and eukaryotic cells. Describe the processes of eukaryotic mitosis and meiosis, and compare to prokaryotic binary fission. Eukaryotic organisms include protozoans, algae, fungi, plants, and animals. Some eukaryotic cells are independent, single-celled microorganisms, whereas others are part of multicellular organisms. The cells of eukaryotic organisms have several distinguishing characteristics. Above all, eukaryotic cells are defined by the presence of a nucleus, surrounded by a complex nuclear membrane. Also, eukaryotic cells are characterized by the presence of membrane-bound organelles in the cytoplasm. Organelles such as mitochondria, the endoplasmic reticulum, Golgi apparatus, lysosomes, and peroxisomes are held in place by the cytoskeleton, an internal network that supports transport of intracellular components and helps maintain cell shape. Figure 3.35. The genome of eukaryotic cells is packaged in multiple, rod-shaped chromosomes, as opposed to the single, circular-shaped chromosome that characterizes most prokaryotic cells. Table 3.2 compares the characteristics of eukaryotic cell structures with those of bacteria and archaea. Figure 3.35, an illustration of a generalized, single-celled eukaryotic organism. Note that cells of eukaryotic organisms vary greatly in terms of structure and function, and a particular cell may not have all of the structures shown here. Summary of cell structures. Cell structure: prokaryotes, eukaryotes. Bacteria: archaea. Size approximately 0.5 to 1 micrometer. Approximately 0.5 to 1 micrometer. Approximately 5 to 20 micrometers. Surface area to volume ratio: high-low. Nucleus: no. Yes. Genome characteristics: single chromosome. Circular. Haploid. Lacks histones. Single chromosome. Circular. Haploid. Contains histones. Multiple chromosomes. Linear. Haploid or diploid. Contains histones. Cell division. Binary fission. Binary fission. Mitosis. Meiosis. Membrane lipid composition. Ester-linked. Straight-chain fatty acids, bilayer. Ether-linked. Branched isoprenoids, bilayer or monolayer. Ester-linked. Straight-chain fatty acids, sterols, bilayer. Cell wall composition. Peptidoglycan or proteinone, pseudopeptidoglycan or ibocyl glycopeptide, or proteinone duller, polysaccharide or ibocyl. Protein S-layer or ibocyl. None. Cellulose, plants, some algae. Chitin, fungi. Silica, some algae. Most others lack cell walls. Motility structures. Rigid spiral flagella composed of flagellin. Rigid spiral flagella composed of archaeoflagellins. Flexible flagella and cilia composed of microtubules. Membrane-bound organelles, no, yes. Endomembrane system, no, yes. Golgi, lysosomes. Ribosomes, 70S, 70S. 80S in cytoplasm and rough 70S in mitochondria, chloroplasts. Table 3.2. Cell morphologies. Eukaryotic cells display a wide variety of different cell morphologies. Possible shapes include spheroid, ovoid, cuboidal, cylindrical, flat, lenticular, fusiform, discoidal, crescent, ring stellate, and polygonal. Figure 3.36. Some eukaryotic cells are irregular in shape, and some are capable of changing shape. The shape of a particular type of eukaryotic cell may be influenced by factors such as its primary function, the organization of its cytoskeleton, the viscosity of its cytoplasm, the rigidity of its cell membrane or cell wall, if it has one, and the physical pressure exerted on it by the surrounding environment and/or adjoining cells. Figure 3.36 eukaryotic cells come in a variety of cell shapes. A. Spheroid cromulina alga. B. Fusiform-shaped trypanosoma. C. Bell-shaped vorticella. D. Ovoid paramecium. E. Ring-shaped plasmodium ovale. Credit A. Modification of work by NOAA. Credit B. E. Modification of work by Centers for Disease Control and Prevention. Check your understanding. Identify two differences between eukaryotic and prokaryotic cells. Nucleus. Unlike prokaryotic cells, in which DNA is loosely contained in the nucleoid region, eukaryotic cells possess a nucleus, which is surrounded by a complex nuclear membrane that houses the DNA genome. Figure 3.37. By containing the cell's DNA, the nucleus ultimately controls all activities of the cell and also serves an essential role in reproduction and heredity. Eukaryotic cells typically have their DNA organized into multiple linear chromosomes. The DNA within the nucleus is highly organized and condensed to fit inside the nucleus, which is accomplished by wrapping the DNA around proteins called histones. Figure 3.37 eukaryotic cells have a well-defined nucleus surrounded by a nuclear membrane. The nucleus of this mammalian lung cell is located in the bottom right corner of the image. The large, dark, oval-shaped structure within the nucleus is the nucleolus. Although most eukaryotic cells have only one nucleus, exceptions exist. For example, protozoans of the genus Paramecium typically have two complete nuclei, a small nucleus that is used for reproduction, micronucleus, and a large nucleus that directs cellular metabolism, macronucleus. Additionally, some fungi transiently form cells with two nuclei, called heterokaryotic cells, during sexual reproduction. Cells whose nuclei divide but whose cytoplasm does not are called coenocytes. The nucleus is bound by a complex nuclear membrane, often called the nuclear envelope, that consists of two distinct lipid bilayers that are contiguous with each other. Figure 3.38. Despite these connections between the inner and outer membranes, each membrane contains unique lipids and proteins on its inner and outer surfaces. The nuclear envelope contains nuclear pores, which are large, rosette-shaped protein complexes that control the movement of materials into and out of the nucleus. The overall shape of the nucleus is determined by the nuclear lamina, a meshwork of intermediate filaments found just inside the nuclear envelope membranes. Outside the nucleus, additional intermediate filaments form a looser mesh and serve to anchor the nucleus in position within the cell. Figure 3.38. In this fluorescent microscope image, all the intermediate filaments have been stained with a bright green fluorescent stain. The nuclear lamina is the intense bright green ring around the faint red nuclei. Eukaryotes are able to multiply through asexual reproduction, during which a single parent cell becomes two identical daughter cells. This process of clonal reproduction is called mitosis. Although mitosis may sound similar to asexual binary fission in prokaryotes, the processes are very different. In contrast to the single chromosome in most prokaryotes, eukaryotic cells possess multiple chromosomes that must be replicated and strategically divided between daughter cells. Therefore, mitosis is a much more complex cellular process than binary fission. The eukaryotic cell cycle is an ordered and carefully regulated series of events involving cell growth, DNA replication, and cell division to produce two clonal daughter cells. One turn or cycle of the cell cycle consists of two general phases, interphase and the mitotic phase. Figure 3.39. During interphase, the cell is not dividing, but rather is undergoing normal growth processes, and DNA is replicated, preparing for cell division. The three stages of interphase are called G1, S, and G2. Figure 3.39. The cell cycle consists of interphase and the mitotic phase. During interphase, the cell grows and the nuclear DNA is duplicated. Interphase is followed by the mitotic phase. During the mitotic phase, the duplicated chromosomes are segregated and distributed into daughter nuclei. The cytoplasm is usually divided as well, resulting in two daughter cells. Attribution, Biology 2E, Rice University OpenStax, under CC by 4.0 license. The mitotic phase is a multi-step process during which the duplicated chromosomes are aligned, separated, moved to opposite poles of the cell, and then are divided into two identical daughter cells. The first portion of the mitotic phase is called karyokinesis, or nuclear division. Karyokinesis is divided into a series of phases: prophase, prometaphase, metaphase, anaphase, and telophase, that result in the division of the cell nucleus. Figure 3.40. The second portion of the mitotic phase, called cytokinesis, is the physical separation of the cytoplasmic components into the two daughter cells. Figure 3.40, karyokinesis or mitosis, is divided into 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase. The pictures at the bottom were taken by fluorescence microscopy, hence the black background, of cells artificially stained by fluorescent dyes. Blue fluorescence indicates DNA, chromosomes, and green fluorescence indicates microtubules, spindle apparatus. Attribution, Biology 2E, Rice University OpenStax, under CC BY 4.0 license. In addition to the mitotic asexual reproduction described above, most eukaryotic microorganisms also have the option of sexual reproduction involving meiosis. Although mitosis and meiosis both require DNA replication, nuclear division, and share procedural similarities, there are important differences between the process and outcomes. Figure 3.41. Figure 3.41, meiosis and mitosis are both preceded by one cycle of DNA replication. However, meiosis includes 2 nuclear divisions. The 4 daughter cells resulting from meiosis are haploid and genetically distinct. The daughter cells resulting from mitosis are diploid and identical to the parent cell. Credit, Biology 2E, Rice University, OpenStax, under CC by 4.0 license. In contrast to the single nuclear division that completes mitosis, meiosis involves 2 separate nuclear divisions. Rather than creating 2 clonal daughter cells, the goal of meiosis is to create 4 genetically distinct gametes, with each gamete possessing half the number of chromosomes found in the original cell. This strategic chromosome reduction is essential for the fertilization that occurs during sexual reproduction to produce in a zygote with a full complement of chromosomes. Nucleolus. The nucleolus is a dense region within the nucleus where ribosomal RNA-rRNA biosynthesis occurs. In addition, the nucleolus is also the site where assembly of ribosomes begins. Pre-ribosomal complexes are assembled from rRNA and proteins in the nucleolus. They are then transported out to the cytoplasm, where ribosome assembly is completed. Figure 3.42. Figure 3.42. A. The nucleolus is the dark, dense area within the nucleus. It is the site of rRNA synthesis and pre-ribosomal assembly. B. Electron micrograph showing the nucleolus. Ribosomes. Ribosomes found in eukaryotic organelles such as mitochondria or chloroplasts have 70 S ribosomes, the same size as prokaryotic ribosomes. However, non-organelle-associated ribosomes in eukaryotic cells are 80 S ribosomes, composed of a 40 S small subunit and a 60 S large subunit. In terms of size and composition, this makes them distinct from the ribosomes of prokaryotic cells. The two types of non-organelle-associated eukaryotic ribosomes are defined by their location in the cell: free ribosomes and membrane-bound ribosomes. Free ribosomes are found in the cytoplasm and serve to synthesize water-soluble proteins. Membrane-bound ribosomes are found attached to the rough endoplasmic reticulum and make proteins for insertion into the cell membrane, or proteins destined for export from the cell. The differences between eukaryotic and prokaryotic ribosomes are clinically relevant because certain antibiotic drugs are designed to target one or the other. For example, cyclohexamide targets eukaryotic action, whereas chloramphenicol targets prokaryotic ribosomes. 24. Since human cells are eukaryotic, they generally are not harmed by antibiotics that destroy the prokaryotic ribosomes in bacteria. However, sometimes negative side effects may occur because mitochondria in human cells contain prokaryotic ribosomes. Endomembrane system. The endomembrane system, unique to eukaryotic cells, is a series of membranous tubules, sacs, and flattened discs that synthesize many cell components and move materials around within the cell. Figure 3.43. Because of their larger cell size, eukaryotic cells require this system to transport materials that cannot be dispersed by diffusion alone. The endomembrane system comprises several organelles and connections between them, including the endoplasmic reticulum, Golgi apparatus, lysosomes, and vesicles. Figure 3.43. The endomembrane system is composed of a series of membranous intracellular structures that facilitate movement of materials throughout the cell and to the cell membrane. Endoplasmic reticulum. The endoplasmic reticulum, ER, is an interconnected array of tubules and cisternae, flattened sacs, with a single lipid bilayer. Figure 3.44. The spaces inside of the cisternae are called lumen of the ER. There are two types of ER: rough endoplasmic reticulum, RER, and smooth endoplasmic reticulum, SER. These two different types of ER are sites for the synthesis of distinctly different types of molecules. RER is studded with ribosomes bound on the cytoplasmic side of the membrane. These ribosomes make proteins destined for the plasma membrane. Figure 3.44. Following synthesis, these proteins are inserted into the membrane of the RER. Small sacs of the RER containing these newly synthesized proteins then bud off as transport vesicles and move either to the Golgi apparatus for further processing, directly to the plasma membrane, to the membrane of another organelle, or out of the cell. Transport vesicles are single-lipid, bilayer, membranous spheres with hollow interiors that carry molecules. SER does not have ribosomes and therefore appears smooth. It is involved in biosynthesis of lipids, carbohydrate metabolism, and detoxification of toxic compounds within the cell. Figure 3.44. The rough endoplasmic reticulum is studded with ribosomes for the synthesis of membrane proteins, which give it its rough appearance. Golgi apparatus. The Golgi apparatus was discovered within the endomembrane system in 1898 by Italian scientist Camillo Golgi, 1843 to 1926, who developed a novel staining technique that showed stacked membrane structures within the cells of Plasmodium, the causative agent of malaria. The Golgi apparatus is composed of a series of membranous discs called dictyosomes, each having a single lipid bilayer that are stacked together. Figure 3.45. Enzymes in the Golgi apparatus modify lipids and proteins transported from the ER to the Golgi, often adding carbohydrate components to them, producing glycolipids, glycoproteins, or proteoglycans. Glycolipids and glycoproteins are often inserted into the plasma membrane and are important for signal recognition by other cells or infectious particles. Different types of cells can be distinguished from one another by the structure and arrangement of the glycolipids and glycoproteins contained in their plasma membranes. These glycolipids and glycoproteins commonly also serve as cell surface receptors, transport vesicles leaving the ER fuse with a Golgi apparatus on its receiving, or cis, face. The proteins are processed within the Golgi apparatus, and then additional transport vesicles containing the modified proteins and lipids pinch off from the Golgi apparatus on its outgoing, or trans, face. These outgoing vesicles move to and fuse with the plasma membrane, or the membrane of other organelles. Exocytosis is the process by which secretory vesicles, spherical membranous sacs, release their contents to the cell's exterior. Figure 3.45. All cells have constitutive secretory pathways in which secretory vesicles transport soluble proteins that are released from the cell continually, constitutively. Certain specialized cells also have regulated secretory pathways, which are used to store soluble proteins in secretory vesicles. Regulated secretion involves substances that are only released in response to certain events or signals. For example, certain cells of the human immune system, e.g., mast cells, secrete histamine in response to the presence of foreign objects or pathogens in the body. Histamine is a compound that triggers various mechanisms used by the immune system to eliminate pathogens. Figure 3.45. A transmission electron micrograph left of a Golgi apparatus in a white blood cell. The illustration right shows the cup-shaped, stacked discs and several transport vesicles. The Golgi apparatus modifies lipids and proteins, producing glycolipids and glycoproteins, respectively, which are commonly inserted into the plasma membrane. Lysosomes. In the 1960s, Belgian scientist Christian de Duve, 1917 to 2013, discovered lysosomes, membrane-bound organelles of the endomembrane system that contain digestive enzymes. Certain types of eukaryotic cells use lysosomes to break down various particles, such as food, damaged organelles or cellular debris, microorganisms, or immune complexes. Compartmentalization of the digestive enzymes within the lysosome allows the cell to efficiently digest matter without harming the cytoplasmic components of the cell. Check your understanding. Name the components of the endomembrane system and describe the function of each component. Peroxisomes. Christian de Duve is also credited with the discovery of peroxisomes, membrane-bound organelles that are not part of the endomembrane system. Figure 3.46. Peroxisomes form independently in the cytoplasm from the synthesis of peroxin proteins by free ribosomes and the incorporation of these peroxin proteins into existing peroxisomes. Growing peroxisomes then divide by a process similar to binary fission. Peroxisomes were first named for their ability to produce hydrogen peroxide, a highly reactive molecule that helps to break down molecules such as uric acid, amino acids, and fatty acids. Peroxisomes also possess the enzyme catalase, which can degrade hydrogen peroxide. Along with the SER, peroxisomes also play a role in lipid biosynthesis. Like lysosomes, the compartmentalization of these degradative molecules within an organelle helps protect the cytoplasmic contents from unwanted damage. The peroxisomes of certain organisms are specialized to meet their particular functional needs. For example, glyoxysomes are modified peroxisomes of yeasts and plant cells that perform several metabolic functions, including the production of sugar molecules. Similarly, glycosomes are modified peroxisomes made by certain trypanosomes, the pathogenic protozoans that cause Chagas disease and African sleeping sickness. Figure 3.46. A transmission electron micrograph left of a cell containing a peroxisome. The illustration, right, shows the location of peroxisomes in a cell. These eukaryotic structures play a role in lipid biosynthesis and breaking down various molecules. They may also have other specialized functions depending on the cell type. Credit Micrograph, Modification of Work by American Society for Microbiology. Cytoskeleton. Eukaryotic cells have an internal cytoskeleton made of microfilaments, intermediate filaments, and microtubules. This matrix of fibers and tubes provides structural support as well as a network over which materials can be transported within the cell and on which organelles can be anchored. Figure 3.47. For example, The process of exocytosis involves the movement of a vesicle via the cytoskeletal network to the plasma membrane, where it can release its contents. A micrograph shows many lines emanating from the nucleus and extending throughout the cell. These are shown in diagram form as small spheres forming the outside of a long tube. Each pair of spheres is a tubulin dimer, and columns of these dimers can be seen on the outside of the large tube they form. The diameter of the tube is 25 micrometers. The same micrograph shows lines throughout the cell. These are drawn as spheres forming abraded structures, a double helix. The diameter of the helix is 7 nanometers. The spheres are labeled actin subunit. Another micrograph shows many lines forming a webbing in the cell. These are drawn as a rope. Each strand of the rope is labeled fibrous subunit, keratins coiled together. The diameter of the rope is 8 to 12 nanometers. Figure 3.47. The cytoskeleton is a network of microfilaments, intermediate filaments, and microtubules found throughout the cytoplasm of a eukaryotic cell. In these fluorescently labeled animal cells, the microtubules are green, the actin microfilaments are red, the nucleus is blue, and keratin, a type of intermediate filament, is yellow. Microfilaments are composed of two intertwined strands of actin, each composed of actin monomers forming filamentous cables 6 nanometers in diameter 25, figure 3.48. The actin filaments work together with motor proteins, like myosin, to affect muscle contraction in animals or the amoeboid movement of some eukaryotic microbes. In amoeboid organisms, actin can be found in two forms: a stiffer, polymerized, gel form, and a more fluid, unpolymerized soluble form. Actin in the gel form creates stability in the ectoplasm, the gel-like area of cytoplasm, just inside the plasma membrane of amoeboid protozoans. Temporary extensions of the cytoplasmic membrane, called pseudopodia, meaning false feet, are produced through the forward flow of soluble actin filaments into the pseudopodia, followed by the gel-sol cycling of the actin filaments, resulting in cell motility. Once the cytoplasm extends outward, forming a pseudopodium, the remaining cytoplasm flows up to join the leading edge, thereby creating forward locomotion. Beyond amoeboid movement, microfilaments are also involved in a variety of other processes in eukaryotic cells, including cytoplasmic streaming, the movement or circulation of cytoplasm within the cell, cleavage furrow formation during cell division, and muscle movement in animals, figure 3.48. These functions are the result of the dynamic nature of microfilaments, which can polymerize and depolymerize relatively easily in response to cellular signals, and their interactions with molecular motors in different types of eukaryotic cells. Figure 3.48a. A microfilament is composed of a pair of actin filaments. B. Each actin filament is a string of polymerized actin monomers. C. The dynamic nature of actin, due to its polymerization and depolymerization and its association with myosin, allows microfilaments to be involved in a variety of cellular processes, including amoeboid movement, cytoplasmic streaming, contractile ring formation during cell division, and muscle contraction in animals. Intermediate filaments, figure 3.49, are a diverse group of cytoskeletal filaments that act as cables within the cell. They are termed intermediate because their 10 nm diameter is thicker than that of actin, but thinner than that of microtubules. 0.26. They are composed of several strands of polymerized subunits that, in turn, are made up of a wide variety of monomers. Intermediate filaments tend to be more permanent in the cell and maintain the position of the nucleus. They also form the nuclear lamina, lining or layer, just inside the nuclear envelope. Additionally, intermediate filaments play a role in anchoring cells together in animal tissues. The intermediate filament protein desmin is found in desmosomes, the protein structures that join muscle cells together and help them resist external physical forces. The intermediate filament protein keratin is a structural protein found in hair, skin, and nails. Figure 3.49a. Intermediate filaments are composed of multiple strands of polymerized subunits. They are more permanent than other cytoskeletal structures and serve a variety of functions. B. Intermediate filaments form much of the nuclear lamina. C. Intermediate filaments form the desmosomes between cells in some animal tissues. Credit C. Illustration, Modification of Work by Mariana Ruiz Villarreal. Microtubules. Figure 3.50, are a third type of cytoskeletal fiber composed of tubulin dimers, alpha-tubulin and beta-tubulin. These form hollow tubes 23 nanometers in diameter that are used as girders within the cytoskeleton. 27. Like microfilaments, microtubules are dynamic and have the ability to rapidly assemble and disassemble. Microtubules also work with motor proteins such as dynein and kinesin to move organelles and vesicles around within the cytoplasm. Additionally, microtubules are the main components of eukaryotic flagella and cilia, composing both the filament and the basal body components. Figure 3.57. Figure 3.50a, microtubules are hollow structures composed of polymerized tubulin dimers. B. They are involved in several cellular processes, including the movement of organelles throughout the cytoplasm. Motor proteins carry organelles along microtubule tracts that crisscross the entire cell. Credit B, modification of work by National Institute on Aging. In addition, microtubules are involved in cell division, forming the mitotic spindle that serves to separate chromosomes during mitosis and meiosis. The mitotic spindle is produced by two centrosomes, which are essentially microtubule organizing centers at opposite ends of the cell. Each centrosome is composed of a pair of centrioles positioned at right angles to each other, and each centriole is an array of 27 parallel microtubules arranged in triplets. Figure 3.51. Figure 3.51a, a centrosome is composed of two centrioles positioned at right angles to each other. Each centriole is composed of 9 triplets of microtubules held together by accessory proteins. B. In animal cells, the centrosomes, arrows, serve as microtubule organizing centers of the mitotic spindle during mitosis. Check your understanding. Compare and contrast the 3 types of cytoskeletal structures described in this section. Mitochondria. The large, complex organelles in which aerobic cellular respiration occurs in eukaryotic cells are called mitochondria. Figure 3.52. The term mitochondrion was first coined by German microbiologist Carl Benda in 1898 and was later connected with the process of respiration by Otto Warburg in 1913. Scientists during the 1960s discovered that mitochondria have their own genome and 70 S ribosomes. The mitochondrial genome was found to be bacterial when it was sequenced in 1976. These findings ultimately supported the endosymbiotic theory proposed by Lynn Margulis, which states that mitochondria originally arose through an endosymbiotic event in which a bacterium capable of aerobic cellular respiration was taken up by phagocytosis into a host cell and remained as a viable intracellular component. Each mitochondrion has 2 lipid membranes. The outer membrane is a remnant of the original host cell's membrane structures. The inner membrane was derived from the bacterial plasma membrane. The electron transport chain for aerobic respiration uses integral proteins embedded in the inner membrane. The mitochondrial matrix, corresponding to the location of the original bacterium cytoplasm, is the current location of many metabolic enzymes. It also contains mitochondrial DNA and 70 S ribosomes. Invaginations of the inner membrane, called cristae, evolved to increase surface area for the location of biochemical reactions. The folding patterns of the cristae differ among various types of eukaryotic cells and are used to distinguish different eukaryotic organisms from each other. Figure 3.52. Each mitochondrion is surrounded by 2 membranes, the inner of which is extensively folded into cristae and is the site of the intermembrane space. The mitochondrial matrix contains the mitochondrial DNA, ribosomes, and metabolic enzymes. The transmission electron micrograph of a mitochondrion on the right shows both membranes, including cristae and the mitochondrial matrix. Credit micrograph, modification of work by Matthew Britton, scale bar data from Matt Russell. Chloroplasts. Plant cells and algal cells contain chloroplasts, the organelles in which photosynthesis occurs. Figure 3.53. All chloroplasts have at least 3 membrane systems: the outer membrane, the inner membrane, and the thylakoid membrane system. Inside the outer and inner membranes is the chloroplast stroma, a gel-like fluid that makes up much of a chloroplast's volume and in which the thylakoid system floats. The thylakoid system is a highly dynamic collection of folded membrane sacs. It is where the green photosynthetic pigment chlorophyll is found and the light reactions of photosynthesis occur. In most plant chloroplasts, the thylakoids are arranged in stacks called grana, singular granum, whereas in some algal chloroplasts, the thylakoids are free-floating. Figure 3.53 photosynthesis takes place in chloroplasts, which have an outer membrane and an inner membrane. Stacks of thylakoids called grana form a third membrane layer. Other organelles similar to mitochondria have arisen in other types of eukaryotes, but their roles differ. Hydrogenosomes are found in some anaerobic eukaryotes and serve as the location of anaerobic hydrogen production. Hydrogenosomes typically lack their own DNA and ribosomes. Kinetoplasts are a variation of the mitochondria found in some eukaryotic pathogens. In these organisms, each cell has a single, long, branched mitochondrion, in which kinetoplast DNA, organized as multiple circular pieces of DNA, is found concentrated at one pole of the cell. Microconnections, mitochondria-related organelles and protozoan parasites. Many protozoans, including several protozoan parasites that cause infections in humans, can be identified by their unusual appearance. Distinguishing features may include complex cell morphologies, the presence of unique organelles, or the absence of common organelles. The protozoan parasites Giardia lamblia and Trichomonas vaginalis are two examples. G. lamblia, a frequent cause of diarrhea in humans and many other animals, is an anaerobic parasite that possesses two nuclei and several flagella. Its Golgi apparatus and endoplasmic reticulum are greatly reduced, and it lacks mitochondria completely. However, it does have organelles known as mitosomes, double membrane-bound organelles that appear to be severely reduced mitochondria. This has led scientists to believe that G. lamblia's ancestors once possessed mitochondria that evolved to become mitosomes. T. vaginalis, which causes the sexually transmitted infection vaginitis, is another protozoan parasite that lacks conventional mitochondria. Instead, it possesses hydrogenosomes, mitochondrial-related, double membrane-bound organelles that produce molecular hydrogen used in cellular metabolism. Scientists believe that hydrogenosomes, like mitosomes, also evolved from mitochondria. Plasma membrane. The plasma membrane of eukaryotic cells is similar in structure to the prokaryotic plasma membrane in that it is composed mainly of phospholipids, forming a bilayer with embedded peripheral and integral proteins. Figure 3.54. These membrane components move within the plane of the membrane according to the fluid mosaic model. However, unlike the prokaryotic membrane, eukaryotic membranes contain sterols, including cholesterol, that alter membrane fluidity. Additionally, many eukaryotic cells contain some specialized lipids, including sphingolipids, which are thought to play a role in maintaining membrane stability, as well as being involved in signal transduction pathways and cell-to-cell communication. Figure 3.54. The eukaryotic plasma membrane is composed of a lipid bilayer with many embedded or associated proteins. It contains cholesterol for the maintenance of membrane, as well as glycoproteins and glycolipids that are important in the recognition of other cells or pathogens. Membrane transport mechanisms. The processes of simple diffusion, facilitated diffusion, and active transport are used in both eukaryotic and prokaryotic cells. However, eukaryotic cells also have the unique ability to perform various types of endocytosis, the uptake of matter through plasma membrane invagination, and vacuole/vesicle formation. Figure 3.55. A type of endocytosis involving the engulfment of large particles through membrane invagination is called phagocytosis, which means cell eating. In phagocytosis, particles, or other cells, are enclosed in a pocket within the membrane, which then pinches off from the membrane to form a vacuole that completely surrounds the particle. Another type of endocytosis is called pinocytosis, which means cell drinking. In pinocytosis, small dissolved materials and liquids are taken into the cell through small vesicles. Saprophytic fungi, for example, obtain their nutrients from dead and decaying matter largely through pinocytosis. Receptor-mediated endocytosis is a type of endocytosis that is initiated by specific molecules called ligands when they bind to cell surface receptors on the membrane. Receptor-mediated endocytosis is the mechanism that peptide and amine-derived hormones use to enter cells, and is also used by various viruses and bacteria for entry into host cells. Figure 3.5. 3 variations of endocytosis are shown. A. In phagocytosis, the cell membrane surrounds the particle and pinches off to form an intracellular vacuole. B. In pinocytosis, the cell membrane surrounds a small volume of fluid and pinches off, forming a vesicle. C. In receptor-mediated endocytosis, the uptake of substances is targeted to a specific substance, a ligand, that binds at the receptor on the external cell membrane. Credit: Modification of work by Mariana Ruiz Villarreal. The process by which secretory vesicles release their contents to the cell's exterior is called exocytosis. Vesicles move toward the plasma membrane and then meld with the membrane, ejecting their contents out of the cell. Exocytosis is used by cells to remove waste products and may also be used to release chemical signals that can be taken up by other cells. Cell wall. In addition to a plasma membrane, some eukaryotic cells have a cell wall. Cells of fungi, algae, plants, and even some protists have cell walls. Depending upon the type of eukaryotic cell, cell walls can be made of a wide range of materials, including cellulose, plants, biogenic silica, calcium carbonate, agar, and carrageenan, protists and algae, or chitin, fungi. In general, all cell walls provide structural stability for the cell and protection from environmental stresses, such as desiccation, changes in osmotic pressure, and traumatic injury. 29. Extracellular matrix. Cells of animals and some protozoans do not have cell walls to help maintain shape and provide structural stability. Instead, these types of eukaryotic cells produce an extracellular matrix for this purpose. They secrete a sticky mass of carbohydrates and proteins into the spaces between adjacent cells, figure 3.56. Some protein components assemble into a basement membrane to which the remaining extracellular matrix components adhere. Proteoglycans typically form the bulky mass of the extracellular matrix, while fibrous proteins, like collagen, provide strength. Both proteoglycans and collagen are attached to fibronectin proteins, which, in turn, are attached to integrin proteins. These integrin proteins interact with transmembrane proteins in the plasma membranes of eukaryotic cells that lack cell walls. In animal cells, the extracellular matrix allows cells within tissues to withstand external stresses and transmit signals from the outside of the cell to the inside. The amount of extracellular matrix is quite extensive in various types of connective tissues, and variations in the extracellular matrix can give different types of tissues their distinct properties. In addition, a host cell's extracellular matrix is often the site where microbial pathogens attach themselves to establish infection. For example, Streptococcus pyogenes, the bacterium that causes strep throat and various other infections, binds to fibronectin in the extracellular matrix of the cells lining the oropharynx, upper region of the throat. Figure 3.56. The extracellular matrix is composed of protein and carbohydrate components. It protects cells from physical stresses and transmits signals arriving at the outside edges of the tissue to cells deeper within the tissue. Flagella and cilia. Some eukaryotic cells use flagella for locomotion. However, eukaryotic flagella are structurally distinct from those found in prokaryotic cells. Whereas the prokaryotic flagellum is a stiff, rotating structure, a eukaryotic flagellum is more like a flexible whip composed of 9 parallel pairs of microtubules surrounding a central pair of microtubules. This arrangement is referred to as a 9+2 array, figure 3.57. The parallel microtubules use dynein motor proteins to move relative to each other, causing the flagellum to bend. Cilia, singular cilium, are a similar external structure found in some eukaryotic cells. Unique to eukaryotes, cilia are shorter than flagella and often cover the entire surface of a cell. However, they are structurally similar to flagella, a 9+2 array of microtubules, and use the same mechanism for movement. A structure called a basal body is found at the base of each cilium and flagellum. The basal body, which attaches the cilium or flagellum to the cell, is composed of an array of triplet microtubules similar to that of a centriole but embedded in the plasma membrane. Because of their shorter length, cilia use a rapid, flexible, waving motion. In addition to motility, cilia may have other functions such as sweeping particles past or into cells. For example, ciliated protozoans use the sweeping of cilia to move food particles into their mouthparts, and ciliated cells in the mammalian respiratory tract beat in synchrony to sweep mucus and debris up and out of the lungs. Figure 3.57. Figure 3.57a. Eukaryotic flagella and cilia are composed of a 9+2 array of microtubules, as seen in this transmission electron micrograph cross-section. B. The sliding of these microtubules relative to each other causes a flagellum to bend. C. An illustration of Trichomonas vaginalis, a flagellated protozoan parasite that causes vaginitis. D. Many protozoans like this paramecium have numerous cilia that aid in locomotion as well as in feeding. Note the mouth opening shown here. Credit D. Modification of work by University of Vermont, National Institutes of Health. Check your understanding. Explain how the cellular envelope of eukaryotic cells compares to that of prokaryotic cells. Explain the difference between eukaryotic and prokaryotic flagella. Clinical focus. Resolution. Since amoxicillin has not resolved Barbara's case of pneumonia, the PA prescribes another antibiotic, azithromycin, which targets bacterial ribosomes rather than peptidoglycan. After taking the azithromycin as directed, Barbara's symptoms resolve and she finally begins to feel like herself again. Presuming no drug resistance to amoxicillin was involved, and given the effectiveness of azithromycin, the causative agent of Barbara's pneumonia is most likely Mycoplasma pneumonia. Even though this bacterium is a prokaryotic cell, it is not inhibited by amoxicillin because it does not have a cell wall and, therefore, does not make peptidoglycan. Go back to the previous clinical focus box. Summary. 3.1 Spontaneous generation. The theory of spontaneous generation states that life arose from non-living matter. It was a long-held belief dating back to Aristotle and the ancient Greeks. Experimentation by Francesco Redi in the 17th century presented the first significant evidence refuting spontaneous generation by showing that flies must have access to meat for maggots to develop on the meat. Prominent scientists designed experiments and argued both in support of John Needham and against Lazzaro Spallanzani spontaneous generation. Louis Pasteur is credited with conclusively disproving the theory of spontaneous generation with his famous swan neck flask experiment. He subsequently proposed that life only comes from life. 3.2 Foundations of modern cell theory. Although cells were first observed in the 1660s by Robert Hooke, cell theory was not well accepted for another 200 years. The work of scientists such as Schleiden, Schwann, Remak, and Virchow contributed to its acceptance. Endosymbiotic theory states that mitochondria and chloroplasts, organelles, found in many types of organisms, have their origins in bacteria. Significant structural and genetic information support this theory. The miasma theory of disease was widely accepted until the 19th century, when it was replaced by the germ theory of disease thanks to the work of Semmelweis, Snow, Pasteur, Lister, and Koch, and others. 3.3 Unique characteristics of prokaryotic cells. Prokaryotic cells differ from eukaryotic cells in that their genetic material is contained in a nucleoid rather than a membrane-bound nucleus. In addition, prokaryotic cells generally lack membrane-bound organelles. Prokaryotic cells of the same species typically share a similar cell morphology and cellular arrangement. Most prokaryotic cells have a cell wall that helps the organism maintain cellular morphology and protects it against changes in osmotic pressure. Outside of the nucleoid, prokaryotic cells may contain extrachromosomal DNA in plasmids. Prokaryotic ribosomes that are found in the cytoplasm have a size of 70s. Some prokaryotic cells have inclusions that store nutrients or chemicals for other uses. Some prokaryotic cells are able to form endospores through sporulation to survive in a dormant state when conditions are unfavorable. Endospores can germinate, transforming back into vegetative cells when conditions improve. In prokaryotic cells, the cell envelope includes a plasma membrane and usually a cell wall. Bacterial membranes are composed of phospholipids with integral or peripheral proteins. The fatty acid components of these phospholipids are ester-linked and are often used to identify specific types of bacteria. The proteins serve a variety of functions, including transport, cell-to-cell communication, and sensing environmental conditions. Archaeal membranes are distinct in that they are composed of fatty acids that are ether-linked to phospholipids. Some molecules can move across the bacterial membrane by simple diffusion, but most large molecules must be actively transported through membrane structures using cellular energy. Prokaryotic cell walls may be composed of peptidoglycan bacteria or pseudopeptidoglycan archaea. Gram-positive bacterial cells are characterized by a thick peptidoglycan layer, whereas gram-negative bacterial cells are characterized by a thin peptidoglycan layer surrounded by an outer membrane. Some prokaryotic cells produce glycocalyx coatings, such as capsules and slime layers, that aid in attachment to surfaces and/or evasion of the host immune system. Some prokaryotic cells have fimbriae or pili, filamentous appendages that aid in attachment to surfaces. Pili are also used in the transfer of genetic material between cells. Some prokaryotic cells use one or more flagella to move through water. Paratrichous bacteria, which have numerous flagella, use runs and tumbles to move purposefully in the direction of a chemical attractant. 3.4 Unique characteristics of eukaryotic cells. Eukaryotic cells are defined by the presence of a nucleus containing the DNA genome and bound by a nuclear membrane, or nuclear envelope, composed of two lipid bilayers that regulate transport of materials into and out of the nucleus through nuclear pores. Eukaryotic cell morphologies vary greatly and may be maintained by various structures, including the cytoskeleton, the cell membrane, and/or the cell wall. The nucleolus, located in the nucleus of eukaryotic cells, is the site of ribosomal synthesis and the first stages of ribosome assembly. Eukaryotic cells contain 80 S ribosomes in the rough endoplasmic reticulum, membrane-bound ribosomes, and cytoplasm-free ribosomes. They contain 70 ribosomes in mitochondria and chloroplasts. Eukaryotic cells have evolved an endomembrane system, containing membrane-bound organelles involved in transport. These include vesicles, the endoplasmic reticulum, and the Golgi apparatus. The smooth endoplasmic reticulum plays a role in lipid biosynthesis, carbohydrate metabolism, and detoxification of toxic compounds. The rough endoplasmic reticulum contains membrane-bound 80 S ribosomes that synthesize proteins destined for the cell membrane. The Golgi apparatus processes proteins and lipids, typically through the addition of sugar molecules, producing glycoproteins or glycolipids, components of the plasma membrane that are used in cell-to-cell communication. Lysosomes contain digestive enzymes that break down small particles ingested by endocytosis, large particles or cells ingested by phagocytosis, and damaged intracellular components. The cytoskeleton, composed of microfilaments, intermediate filaments, and microtubules, provides structural support in eukaryotic cells and serves as a network for transport of intracellular materials. Centrosomes are microtubule organizing centers important in the formation of the mitotic spindle in mitosis. Mitochondria are the site of cellular respiration. They have two membranes, an outer membrane and an inner membrane with cristae. The mitochondrial matrix, within the inner membrane, contains the mitochondrial DNA, 70 S ribosomes, and metabolic enzymes. The plasma membrane of eukaryotic cells is structurally similar to that found in prokaryotic cells, and membrane components move according to the fluid mosaic model. However, eukaryotic membranes contain sterols, which alter membrane fluidity, as well as glycoproteins and glycolipids, which help the cell recognize other cells and infectious particles. In addition to active transport and passive transport, eukaryotic cell membranes can take material into the cell via endocytosis, or expel matter from the cell via exocytosis. Cells of fungi, algae, plants, and some protists have a cell wall, whereas cells of animals and some protozoans have a sticky extracellular matrix that provides structural support and mediates cellular signaling. Eukaryotic flagella are structurally distinct from prokaryotic flagella, but serve a similar purpose, locomotion. Cilia are structurally similar to eukaryotic flagella, but shorter. They may be used for locomotion, feeding, or movement of extracellular particles.